Hello, I will be discussing the Sonic Hedgehog signaling pathway and its role in creating the fingers and thumbs on all of our hands. Sonic Hedgehog is a segment polarity protein that governs the anterior-posterior polarity of the body. When looking at an embryo, humans start off with a fin-like structure called a bud, where the hand should be. Let's look a little closer at the bud. There is a region on the posterior portion of the bud known as the zone of polarizing activity, ZPA for short. This region of mesoderm cells produces the Sonic Hedgehog protein, which creates a concentration gradient across the bud. The release of Sonic Hedgehog triggers the formation of the fingers and the thumb. Now let's talk more about the Sonic Hedgehog pathway and how it works. Sonic Hedgehog occurs in the primary cilia. The inactivated state has the protein patched in the cell membrane, and it is bound to the protein smoothened, which is suppressed when bound to patched. Smoothen remains in the cytoplasm until it is activated, where it then moves to the plasma membrane. Next you have Gli proteins 1, 2, and 3. We'll just be referring to Gli3 in this example. The Gli proteins are associated with a SUFU protein, which is another suppressor protein. Proteins CPI, PKA, and GSK3 beta phosphorylate Gli3. After the phosphorylation of Gli3, the protein is cleaved, creating Gli3R. Gli3R travels to the nucleus, where it binds to the DNA and inhibits the transcription of the genes that require Sonic Hedgehog for activation. When Sonic Hedgehog is present, it binds to the patched protein, and the cell is now in an activated state. The binding of Sonic Hedgehog to patched causes patched to become unassociated with smoothened. Patched is then degraded by proteasomes. Smoothened is now activated and becomes phosphorylated by the proteins GPRK2 and CPI. It can now enter the plasma membrane. Unlike the inactivated state, Gli3 does not become phosphorylated by CPI, PKA, and GSK3 beta. Gli3 then dissociates with SUFU and binds to KIF7, which aids Gli3 in transporting through the cilium. Gli3 then travels to the nucleus, where it activates Gli target genes on the DNA. For this cell type, the activation of the Gli target genes would signal the surrounding tissue cells to begin separating into fingers and a thumb. Sonic Hedgehog is responsible for many things during development. Some examples are creating a midline in the body, limb development, organ and teeth development, neuron development, and developmental patterning. Some examples of birth defects caused by the overproduction or the absence of Sonic Hedgehog are cyclopia, extra fingers or missing fingers, holoprosencephaly, and schizophrenia.